Human Genome Project. In the preceding sections, you have learned that it is the sequence of bases in the DNA that determine the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different at least at some places. These assumptions led to the of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining the DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. So, Human Genome Project was called as a mega project. You can imagine the magnitude and the requirements for the projects if we simply define the aims of the project as follows. So, human genome is said to have approximately 3 into 10 to the power 9 base pair, and if the cost of sequencing required is 10 US dollars per base pair, the estimated cost in the beginning, the total estimated cost of the project would be approximately 9 billion US dollars. Okay, so further, if the obtained sequences were to be stored in typed forms in books, and if each page of the book contained 1,000 letters and each book contained 1,000 pages, then 3,300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. The Human Genome Project was closely associated with the rapid development of a new area in biology, that is bioinformatics. So, goals of Human Genome Project. Some of the important goals of Human Genome Project were as follows. It, it aimed to identify all the approximately 20,000 से 25,000 genes in human DNA, determine the sequence of the 3 billion chemical base pairs that make up the human DNA. Store this information in databases, improve the tools for data analysis, transfer the related technologies to other sectors such as industries, address the ethical, legal, and social issues, means LC, that may arise from the project. So, the Human Genome Project was a 13-year-old project coordinated by the US Department of Energy and the National Institution of Health. During the early years of the Human Genome Project, the Wellcome Trust of UK became a major partner. Additionally, additional contributions came from Japan, France, Germany, China, and others. The project was completed in 2003. However, knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect human beings. So, besides providing the clues to understanding human biology, learning about non human organisms' DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non human model organisms such as bacteria, yeast, C. elegans means Caenorhabditis elegans, a free living non pathogenic nematode. Drosophila, the fruit fly plants, rice, and Arabidopsis, etc., have been sequenced. Methodologies. The methods involved in the involve two major approaches. One approach focused on identifying all the genes that are expressed as RNA, means expressed sequence tags. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequences, and later assigning different regions in the sequence with functions, a term referred to as sequencing sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes and cloned in suitable host using specialized vectors. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria, yeast, and the vectors were called as bacterial artificial chromosome and yeast artificial chromosomes. So the fragments were sequenced using automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. Remember, Sanger is also credited for developing the method for determination of amino acid sequences in proteins. So these sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, 22 autosomes, and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites. One of the application of polymorphism in repetitive DNA sequencing shall be explained in next section of DNA fingerprinting. Now the salient features of human genome are that the human genome contains 3164.7 million base pairs. The average gene consists of 3000 bases, but sizes vary greatly, with the largest human gene being dystrophin at 2.4 million bases. The total number of genes is estimated at 30,000, much lower than the previous estimate of 80,000 to 140,000 genes. Almost all, means 99.9% nucleotide bases are exactly the same in all people. 0.1% ही vary करता है, and that 0.1% makes every person unique. The functions are unknown for over 50% of the discovered genes. Less than 2% of the genome codes for proteins. Repeated sequences make up large portion of the human genome. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has many genes, 2968, and the Y has the fewest, 231. Scientists have identified about 1.4 million locations where single base DNA differences, SNPs, single nucleotide polymorphism, pronounced as SNPs, occur in humans. So, this information promises to revolutionize the processes of finding the chromosomal location for disease associated sequences and tracing the human history. So, the applications and future challenges deriving many meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems. The enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impact of having the human genome sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or a few genes at a time with whole genome sequences and new high throughout, throughput technologies. We could approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life.